I do know the feeling. Yeah.
I always have a sweater with me since I'm in the US in the summer because when I go inside it's freezing. Anyways, okay, I think we can start. So welcome everyone to Science Society, and a special welcome to Georgia. I probably say your name wrong.
No, no,
Georgia.
it's perfect. It's perfect,
Georgia.
Georgia.
Okay. And let me introduce you our guest speaker today a little bit so you get to know her. Georgia de la Ferreira is a PhD student at IBM. She joined the IBM Research Zurich Lab in 2020 as a PhD student in neuromorphic computing and groups, and she's participating in a joint PhD program between the Swiss Federal Institute of Technology and the University of Zurich and the IBM Research Zurich Lab. And during her PhD, she has been working as a visiting student at the Harvard Medical School in the group of Professor Kreiman. And Georgia's current research focuses on developing biologically inspired training algorithms for artificial and spiking neural networks. She is also excited about applying machine learning models to predict and guide neural activity in the animal brain. And Georgia did her master's in applied physics from the École Polytechnique Fédérale de Lausanne and has served as a research intern at the Okinawa Institute for Science and Technology in Japan, Logitech, the Swiss Space Center, and Imperial College London. So in her life, she did already so much. And in her free time, if you believe it, she has a little bit of free time. She has worked as a dancer and a dance instructor. That's wonderful. That's really great. I love when people also have their creativity.
Thank
And
you.
if it's okay with you, Jamie will ask you first like a couple of general questions, and then it's time for you to present your amazing work. So go
Yes,
ahead,
that's
Jamie.
perfect.
Thank you.
Thank you very much, and thank you so much for joining us, Georgia. And that is an incredible bio there, I have to say. And I'd like to
Yes.
start off just by asking you, please, what is it that first of all got you into science? What fascinated you? Any stories you could remember on where you first found all this kind of stuff fascinating?
Yes, sure. Actually, when I was in high school, I chose to do a humanistic high school because I wanted to go towards history or philosophy. But then when I was doing that, I did a project in astrophysics, and that's where I decided I want to do physics. And then as I was doing physics, I had the chance to do a minor in neuroscience, and I really like neuroscience. So it's a non super linear choice, but science has always been there. And I'm really happy now I've landed into computational neuroscience because I get to see both world of like programming and modeling and like more biological knowledge and try to bridge the gap, at least give a small contribution towards that.
That's absolutely fascinating, and this is my curiosity too. What's about the dancing? What got you into that? Is that your
Dancing,
family
my
thing?
father actually.
Oh,
No, I started when I was like 3 years old, so it was not really my decision. But I've continued and I'm still doing it now, and it's really nice because I have the scientific part of the day and then the more artistically part of the evening. So it's a good balance, but I still haven't found a way to merge the two. But we might work on a project with a choreographer in Zurich, try to apply AI models to choreographing in contemporary dance. but it's still like at the brainstorming level. I'm really excited about it, but we'll see where it gets us.
That really does sound exciting. And so, what is the path that you took that led you up to the paper that you'll be discussing with us today?
So you mean this specific project?
Yeah. What is it that got you into this particular one? Yeah.
Yeah, so when I finished my master's, I had a few months gap before starting my PhD, and I wanted to combine doing neuroscience with traveling. So I went to Japan for an internship. I had read the papers of Professor Fukai, who's the last author of the paper. And so I contacted him, invited me to join his lab for a few months. And I was working when I was in Logitech on speech processing with spiking network. And Professor Fukai had developed a new learning rule for spiking network. So we tried to combine the two aspects. Between like my knowledge in audio and his new algorithm. And this is how the paper came out.
That's actually incredible. I'm looking forward to this now. Thank you so much for giving us those answers and giving us a little bit of insight into what got you here. And with no further ado, please, Georgia, the stage is yours.
Thank you for the question, and thank you for the very nice introduction. So I have shared the link with the slides. I put some slide numbers, so I hope you can follow me easily. I'll try to remember every time I change slide to say it. But please, if anyone is lost or confused, stop me, and then I'll try to put everyone again on the same page. And then if there is anything unclear, ask questions during the presentation. I'd really love it to be an interactive discussion. So I'll start with the first slide, which has the title, just to give a very quick overview of the points we're going to touch on during the presentation. So the idea is to do recovering blind sources through mixture repetition. Blind sources are generally sounds or visual stimuli that can be mixed. And the idea of the paper is to find a way through repetition of these mixtures to recover them. So to infer what were the original signal sources before they were mixed. And then, as I briefly mentioned, the PI of the investigation is Professor Fukai, who's now at the Okinawa Institute for Science and Technology. And also we work with Toshitaka Zabuki, who's he was a postdoc there at the time. Now he just moved to London. And you can see that this paper has been published just a couple of months ago, but actually we worked on it probably 3 years ago now, and then it took a while to get the manuscript as a side project into the final version. So I hope I still remember all the details if you want to know very specifically what you did, but please don't be shy. So starting with the second slide, a bit on the motivation of the project. So I mentioned that we are working with a new learning rule that was developed in Fukai's lab, and that is done specifically to train artificial dendritic neurons. So we're talking about spiking networks, which are a subset of artificial neural networks in which the node, which are the basic components of the network, describe or try to mimic the dynamics of biological neurons. There are different ways of doing this, different types of spiking neurons. You may have heard about the leaky integrate and fire neurons, which are just one compartmental model, which means that the neuron is described basically just by one value, which is the voltage threshold, and then by the binary behavior of spiking or not. But we're using a slightly more complex model that you see here in the leftmost panel, which is two compartmental. We have, as you can see, two sort of units that compose the neuron. The top one in the blue square that you see denoted as V is the dendritic compartment. and basically represents the activity that the dendrites of the neuron are integrating from the presynaptic population or from the input signal, for example. And then from the dendrites, this signal travels through the soma, so from the top square to the bottom round part, which is the body of the neuron, and here is denoted as U. And by comparing the activities of the dendrites and the soma, and seeing if there is any mismatch among the two values as the neuron dynamic evolves, the weights that you see denoted as W in orange at the top are evolved. So I will not go into the details of the equations, but the main points that you should keep in mind is that this rule is self-supervising. There is no explicit error signal, and then has been specifically developed to learn temporal features. It has been previously applied to a wide variety of tasks, and also in another context and with other settings, it has shown to be able to perform blind separation of signals, where, for example, the signals are melodies played by two instruments at the same time. And then if we look at the right part of the slide, what we want to apply this learning rule, this model to, is an experiment that was proposed in 2011 by Professor McDermott, who's now at MIT, and that was published in PNAS, so about 10 years ago. It's quite an old experiment, where the idea is to do some blind source separation, so separating signal sources, in this case acoustic sources, through repetition. So the idea is, if we look at the left part of this right quadrant, is that we're going to mix signals. For example, we mix the sound of a trumpet with the sound of a train, and then we present it to an observer. And then we mix the same sound of the trumpet with the sound of an elephant, and we present it to a listener. And then this is what we can call a sort of training phase. And then we ask the listener to classify some sounds as sounds that have been previously played during the mixtures or not. So for example, if we present the sound of the trumpet, the listener has to say, yes, it was present in the mixture. If we present another instrument, the listener has to distinguish that this is an unseen sound. So this is one way that we can perform blind source separation. But let's now go a bit in detail on what source separation is. So if we go on slide 3, we'll just get a quick overview of what I'm going to talk about. I will first give a brief overview of blind source separation in general and about the experiments that we're trying to model with the dendritic neurons. I'm going to give a bit of details on the network architectures, and then present the results on 3 experiments on synthetic sounds that I'm going to explain how they are generated, and then our extra results on real-world stimuli that are an extension of the original experiments. So let's start with the experiments with human listener. If you go on slide 5, you can see an example of how blind source separation can be illustrated. It is often referred to as the cocktail party problem. In short, you have different sound sources that you can see here, for example, as a man singing, a man playing an instrument, a man playing a trumpet. And then all these sounds are mixed together and recorded by some instruments such as microphones. And the problem of blind source separation is decoding the original sources. So given the overlapped signals, you want to separate what signal is the singing, what signal is the violin, and so on. And if you go on slide 6, we can see one of the solutions among the many ones that have been proposed in the past few years. So it has been shown by McDermott and colleagues that humans can perform this blind source separation task by identifying sounds as patterns that are occurring recurrently in mixtures. So we take, for example, two scenarios. If you look at scenario A at the bottom in the train section, you see that we have one target sound, the red sound, which is always repeated in the mixture with the same sound in blue. And in this case, if we only hear the target sound, so for example, it was the trumpet in our initial example, always in combination with the same sound, for example, the elephant, at the end, the listener cannot recognize if it has heard the sound or not, because it always relates it to the other sound. However, the discovery was if you present the sound in different mixtures, so this would be the case B, in which a training sequence contains the target sound, the red one, combined with two different masker sounds, the blue and the green, then after listening to these multiple mixtures, the human listener is able quite confidently to say if the test sound was present or not. So you can see in the test column that we have two types of sounds that are tested. Either the red one that you see during the train, which is the target sound, or depicted with red with a black contour. It is what we call the distractor. So a distractor sound is a sound that is very similar to the target, but is not the same sound. So we want to make the task difficult for the listener and present a sound that is similar to what it has heard, but not identical. And then the listener has to distinguish that this is an unseen sound. So if we go to slide 7, you can see the types of sound that we're dealing with. We cannot use for this experiment sounds that are naturalistic because we have already heard sounds previously in our life. So if we present a listener with the trumpet sound, it is not the first mixture that the listener has listened the trumpet in. So the experimenters needed to create a new data set of sounds. And to do so, they basically looked at a huge number of natural sounds. Based on their features, they built correlation functions. And from these correlation functions, they defined some Gaussian distribution, 5 in total, that combined with some noise can generate new sounds. In particular, they used 5 Gaussian distributions, which correspond to the 5 rows of the images that you see, and then 10 types of sounds per Gaussian distribution, which would correspond to the columns. So these sounds, as you can see, are very similar across columns. So for example, if you look at the first row, all the columns have some time frequency pattern that is scattered around this is called a spectrogram, this visual representation of the sound. While if you look at the bottom row, you have that there are some specific time frames at which the sound is high intensity, so it's yellow, and this is spread all across the frequency level. So in this way, 50 different sounds were generated. And then if you go to the next slide, you can see how the distractor sounds are built. So the ones that we want to test the listeners on. So basically, let's say that our target is in panel A, and then we want to test the listener to another sound that is similar. We basically sample another sound from the same Gaussian distribution, so from the same row among those that I've shown in the previous slide. And then to make the task even a bit harder, you randomly select one time slice, which is equal 1/8 of the total length of the sound. And then you substitute this time slice with the same value that you would have in the target. So now we have the basic units that we need for the experiment. So the target sounds that we are going to mix with some masker sounds, and then the distractor sounds that we need for the test. Is anything unclear for now, or should I go on?
For me, it's fine, but yeah, please flash your microphones if you have any questions so far.
Okay, perfect. At least I have a feedback that you've heard me so far.
I think, Maya, do you have a question?
Yeah, actually, I do. Let me know if we could talk about this, maybe a little bit later along in the presentation. But are you familiar, what's your familiarity level with ultrasonic waves to kind of go through pipes and stimulate, utilizing frequencies either to clean pipes or to put sound into, let's say, a fluid, which could then kind of heat it up, cause pressure. Are you familiar with that kind of technology?
No, not really. So are you bringing it up because you think it would be a suitable sound source?
Well, I'm just bringing it up to understand, the high frequency ultrasound capabilities to use as, potential energy conversion sources.
Maya, this is a neuromorphic or the topic is if you see about developing newer, better AI versions. So it's really kind of off topic.
Got it.
But
Got it. That's what I was asking. I didn't know where we were at, but thanks for the clarification.
Okay, thank you. Thank you for the question.
I have a quick question.
Sure.
If it's okay here, you mentioned in your paper the blind source decomposition problem. If you're going to address this a bit later on, I can wait for my question for later on, because I know that you're in the middle of something. Is it okay to ask about that just now?
Yeah, of course.
You mentioned the blind source decomposition problem. Does that include something like hearing a car coming, like in a city? You can't tell which direction it's coming from because the sound's bouncing against everything.
So it is more having different sound types and wanted to distinguish them. So
Ah,
from your example, maybe you have the car sound, but then you have, I don't know, a street musician playing nearby. And so you have the two sounds that are mixed. And you kind of want to understand that the overall sound is composed of two different sources, the car sound and then the musician sound, and you want to separate them. This is the overall problem.
right. Because that actually was one other quick thing then is isn't part of what makes it difficult to distinguish a sound is that one sound can like supersede and even drown it out? Like, do they still keep being two sounds if they both have the same sort of similarities? Can that happen?
Sorry, can you just repeat the last sentence?
Sure. Is part of what makes it difficult to distinguish these two sounds, is if one sound can supersede another sound and drown it out,
Mm-hmm.
kind of corrupting the sound, making it difficult to hear in that sense? Or does the two sounds keep existing, or can one write over the other sound?
Yeah, so I would say if you want to go into the simplest scenario, you would have the two sounds that have more or less the same intensity. So you don't have one that is very, very loud and one that is just a whisper, because you want to make the task fair. Then, of course, this is not reality. This is just AI benchmark to make sure that you're just narrowing down to the key factors. And then, of course, if you have sounds that are very similar, it's going to be more complex to separate them. But what we're dealing with for these specific projects are these sounds that are synthetic. They are artificial, so they're not sounds that you would hear in the everyday life. And this is done on purpose, first of all, because you don't want familiarity, but also to remove all these factors that are due to the nature of the sound or or like some more overwhelming presentation of a sound compared to another.
Ah, okay. Thank you very much for answering. Thank you.
Okay, good. Thank you very much for the question. Okay, so I hope now the idea of the experiment that has been done in human is clear. Just again to shortly summarize, humans are shown with sounds in mixtures, and then mixtures can be just one or multiple, and then they have to recognize in a test phase if within this mixture they have heard the testing sound or not. And the aim of the project is to see if we can reproduce this experiment by using a very simple spiking model. So if you go to slide 10, you're going to see the model that we came up with. It is a very, very simple model of the acoustic system, and it's based on spiking neurons. So looking at the pipeline, you start with a sound that you depict as a 2D image, which would be the spectrogram. The x-axis is the time, the y-axis is the frequency. So basically this is a matrix of numbers with an intensity that we normalize from 0 to 1, for example. And to make the task a bit easier, we flatten this 2D matrix into one vector. So basically we take each column of this image and then we stack the column on top of the others. And this would be the vector that you see in the middle figure. And then, so you're going to have n pixels. And you build a network so that the number of input neurons is also n. So for each pixel of the spectrogram, either its 2D version or its flattened version into a vector, you associate one neuron. This neuron is called Poisson neuron because it follows a Poisson distribution for spiking. It's a probability distribution. Just in a simple way, if a neuron is associated to a pixel that is very bright, so it's very intense, it is going to have a high firing rate. This means that at each time step, its probability to spike is high. Conversely, if one neuron is associated to a purple pixel, so a low intensity pixel, its probability to spike will be low. So when we want to present an image to a spiking network, we have to convert it into spike. Basically, we present the image for a number of time steps that we define to be 400. And then for these 400 time steps, each neuron is going to spike with one probability defined by the pixel intensity. And this will be the activity of the input layer, the one that you see with the white dots on the left. And then these these spikes, travel through the connections in gray, the ones that are called afferent synapses, excitatory or inhibitory. And then the signal travels through the output layer, which are the nodes on the left part of the picture, that are instead the dendritic neurons. So coming back to the question I had at the very beginning, that was, don't all neurons have dendrites? So in biology, yes. But as you can see, in this input layer, we have Poisson neurons that are just spiking or not. So their activity is a binary value. We don't have any complex information about what is dendrite and what is soma. It's either 0 or 1. Instead, the dendritic neurons that we have at the output are have these two compartmental structure in which the information from the input nodes will first flow to the dendrites V and then propagate to the soma U. And based on the learning rule that was proposed by Professor Fokai, we train the weights that connect the input to the output node so that the network can perform our task. There are also other connections that make the picture a bit more complex, which are these light blue arrows that you see on the output layer. And these are lateral synapses that are inhibitory. Basically, we want the activity of the output neurons to be different across neurons. We want one neuron to spike very strongly as a response to one input, but not one other input. And we want another neuron to have a different behavior so that the dynamics is non-trivial. So just to summarize, if you go to slide 11, so the architecture is just this two-layer network of spiking nodes. The connections are either all-to-all, this fully connected option, or sparse, which means that only 30% of the weights are non-zeros. The number of input neurons is the same as the number of input pixels, so that we can perform this rate encoding. And then in the output layer, we investigated different number of neurons. This number doesn't change much in the results, but in the picture plots that I'll show later, for the sounds we use 8 neurons, and for the images we use 5. So
can I ask a couple of questions?
Sure.
So you were saying this is fully connected. Does that mean that you talked about inhibitory neurons in the output layer? Are all of them fully connected with the first layer, and where is the inhibition targeted? Is it targeting the dendritic compartment or the somatic compartment in the outer layer?
Okay, sure. So we have all-to-all connectivity from the input to the output neurons. So these would be the gray lines that you see in the image. And this can be both excitatory and inhibitory, meaning that the weights are both positive and negative. We initialize with a mean zero distribution, if I remember correctly. And then when I say that this can be either all-to-all connectivity or sparse connectivity, I'm talking about these gray weights. When I mention the lateral inhibition, basically you have from here in the picture, if you look, we have all the connections originating from the 4th neuron from the top and going to all neurons that are neighboring. Well, besides, I don't know why there's one arrow missing for the last neuron, but basically you have this connectivity that is unidirectional, and then this is from one neuron, and of course, just for illustration purpose, we put one, but you could put for all the other neurons. And the inhibition, I don't remember exactly, but I think it targets the dendrite because it's where you integrate the information from, and you don't want it to spike because you want the soma to reproduce what the dendrite is doing, and you want the dendrite to be silent if the neighboring neuron is spiking.
And this is all feed forward, or is there any feedback signals coming back anywhere? All feed forward. And yes. It's unclear to me, besides inhibition, lateral inhibition, what is targeting the dendrites? Are the feed forward connections targeting dendrites and somatic compartments, or?
I see. So they are targeting the dendrites. So as you can see in the dendritic neuron square, you have the weights that reach the V, so the dendrites. And then basically what the soma is doing is trying to reproduce the dynamics of the dendrites. And if there is a mismatch, so if the soma is not well trained yet, then there is a change in plasticity. If there is not, so if the neuron has been trained well, because we are talking about temporal processing, we have for 400 time step the presentation of the same input, so that the soma has enough time to learn how to follow what the dendrite is doing. And this gives the possibility,
Sorry, go ahead.
And this gives the possibility of learning these weights between the input nodes and the dendritic compartment of the output nodes.
Awesome. So last question, and I don't want to derail you anymore. We can always set this aside and talk later. I am curious about the learning rule, though. Is it similar to Walter Sen's work, where we're talking about dendritic predictive coding, or the basically the cells trying to equilibrate the neuron, the dendrites trying to predict the somatic compartment and this kind of thing. And we use that to drive, well, in this case, there's just bottom-up synapses, right, in the somatic compartment.
Yeah, so to be honest, I don't remember how Walterson's predictive rule works. I maybe because I didn't prepare backup slides that are so technical, but basically it is somatodendritic mismatch minimization. So you compute the KL divergence, and this is what you want to minimize.
Yeah, it's the same concept. That makes a lot of sense. Thanks.
Okay, perfect. Thanks for the questions. Okay, I'll go ahead then to the experiments on the original data sets. So if you go on slide 13, you have the details of how the training works. If you remember, what we are presenting the human listeners with are these mixtures. Actually, in the human experiments, the listeners are presented just once with the mixtures, and then they are tested. While in our experiment, we need a bit longer training process, which means that we build a mixture. We can see both the A or the B cases on the top right of the slide. And each sequence is presented 3,000 times. Basically, if you remember, the mixtures, so the spectrograms of the mixtures are encoded into spiking. Then we let the dynamics evolve, and the learning rule trains the weights. And you can see in the bottom right plot how the weights change as we present the train sequence. So you can see that the delta W, which is the magnitude of the weight change, is pretty high at the beginning of the training, and then after about 1,200 iteration, there is already saturation. So the weights have been learned already. Going back to the learning rule, so it's basically a sort of Hebbian learning rule, which means you have pre and post synaptic information for the weight change, and then you have backpropagating action potential. So this is what I was mentioning. You have a sort of prediction attempt from the soma that wants to learn what the pattern that the dendrites are encoding as activity is. And if you remember from the architecture slide, we have this light blue arrow that represent the lateral inhibition that gives rise to the competition across the neurons, so that each neuron responds differently to the same input. And this is implemented through ISTDP, so inhibitory spike time independent plasticity, which is a learning rule that is based on the time difference of the spike of pre and postsynaptic nodes, and in this case, lateral connected neurons. So, to keep in mind, we present the network with these mixtures for 3,000 times, and then we test it. So now it's slide 14. In the test, we present instead the testing sequence, which is composed by single sounds, not mixtures anymore. And the single sounds are the targets and masking sounds, which can be the red, light blue, and green colors, or in the picture even the yellow. And then we present the distractor sounds that, if you remember, are sounds that are similar to the sounds composing the mixtures, but are a bit different. And we want to detect if the network is responding differently, for example, to the red versus the red and black sound. And I mentioned we have 8 output neurons for the sound task. And you can see for each row, the activity of each neuron in terms of firing rate as a response to the different sounds during the test sequence. And this is one presentation. Then we present it 50 times, and then again 20 times in a loop, so that we have a nice averaging, because you can see that there is some oscillation. So how do we go from these population response, in which we see how each neuron responds to a sound, to the classification that we did with human subjects, so saying if the sound was in the mixtures or not. In slide 15, we see the first step of going from the activity to the final response, which is basically applying just simply PCA to reduce the dimensionality of the response. So when we have 8 neurons, each data point is 8-dimensional, which is quite difficult to visualize. So we go back to 2 dimensions and apply PCA to the responses. Here you see for each point the response to the target and the masker sound. So the red, light blue, green, and yellow sound. And each point color corresponds to one of the sound IDs. You see that points corresponding to the same sounds are close in space. This means that the response is similar, but there is some variability. Indeed, if you remember, all the encoding process relies on a Poisson process, so there is some noise within the encoding. Then we go to slide 16, and we perform the next step. So based on the PCA that we've done in the previous step, now we apply a Gaussian mixture model to locate in the PCA space some Gaussian distributions that describe the targets and masker sounds, so the sounds that have been presented during training, which are now represented all in blue. And as you can see, the contour lines of the Gaussian mixture model follow accurately the clusters produced by the targets. Then we plot on this space the response of the distractors, which are in green. And some of them are overlapping with the targets, but most of them, as you can see, are far away from the clusters in between. So being close or far from the clusters is the measure that we call log likelihood. And when the contour lines are dark, the log likelihood is high, close to 1, so the purple areas. When the log likelihood is low, so the probability of one point to belong into one cluster is low, then we have this orangish color. And ideally, our aim is that if the model is able to distinguish targets from distractors, then the distractors will not fall close to the targets in the GMM. So if you go to slide 17, we basically divide, just empirically, the range of the log likelihood into some bins that are corresponding to one answer per point. So if the log likelihood of a point is larger than 0, it means that the point is in the cluster, and the network is saying, yes, this data point, this sound, has been presented in the training mixtures. If at the opposite, the log likelihood is very, very small, so smaller than -15, the network is saying, no, this point is very far away from the clusters that we defined with the target, so this is a distractor. And the answer is sure no. And then the intermediate ranges are yes and no. And at this point, we have exactly the same information that the human subject provided us, because also the human subjects were saying sure yes, yes, no, or sure no. And based on the responses, so you can go now to slide 18, we build a ROC curve. And slide 19, we compute the area under this curve. So if the classification is perfect, we are basically going to have a vertical line and then an horizontal line. So more or less what we have in this figure. And the area under the curve is 1, so the performance is perfect. No distractor is classified to be close to the targets, and all the targets are close enough to be within the clusters. If instead the distractor points fall on top of the target, then we are going to have a line which is diagonal, just 2 points, so going from 0, 0 to 1, 1, and therefore it's just the lower part of a triangle, and this is 0.5, the area under the curve, and this corresponds to the chance level. So basically, just summarizing these slides, after training, we freeze the weights, we compute the network response to the targets and the distractor sounds. We build a PCA space based on the targets. On the PCA space, we apply the Gaussian mixture model to define the Gaussian distributions. On top of the target space, we project the distractor responses. And then for targets and distractors, we compute the log likelihood. And based on this classification, we obtain a ROC curve. And if the network is able to distinguish target and distractor, we obtain area under the curve 1, otherwise we obtain 0.5. So if there's any question, I think this is a good moment. Otherwise, I'll go to the results. Okay, I'll go to the results. So the first experiment that we tried is varying the number of mixtures. So you can see on the left quadrant, we have 3 cases. One mixture only, so we always have the red sound in combination with the blue one. Then, and here the experimental results have shown that the human cannot distinguish targets from distractors. Second case, we have 2 mixtures, so 2 masker sounds. Third case, we have 3 masker sounds and 3 different mixtures. We extended this towards the experiment 1 all combination protocol, in which in addition, we also present during training the mixtures between the other masker sounds, so like the blue with the green. And on this protocol, then we tested the model. Now is slide 21. We look at the network response when we only train it with 1 mixture. And you see in panel A that the response is quite oscillating, but very close independently of the sounds. So then when we build the PCA space, we only have the 2 sounds, which are 1 and 45, you can see, and they all fall in the same cluster. And then when we put the GMM and the distractor sound on top, which would be panel C, the orange and red colors, this falls all on top. So basically targets and distractors are not distinguished by the network. And you see, because the activity in panel A is very similar independent of having correct or incorrect probes, which is another way of saying target and distractor. But if you now move to slide 22, in panel D, you see the response after the network has been trained with 3 mixtures. And you see now that the activity is very pronounced for some neurons and some specific sounds. For example, neuron 1 spikes a lot as a response to the sound in blue, neuron 2 as a response to sound in green, and so on. And these responses are different when we present the distractors. So now if we apply the same pipeline and we look at what happens in panel F, you see that some distractors are indeed confused with the targets, but many others are actually classified as away from those clusters. So to summarize the performance, slide 23 now, what we are interested in is having 1 mixture that is chance level, which is what happens in human performance. If you look at the right histogram on the right. And what we see is that with our dendritic model, the performance is also close to 0.5. When we increase the number of mixtures, like in the human experiment, you have an above chance accuracy. The limitation of our model is that after 6 mixtures, however, the accuracy goes back to chance level, which is probably due to the fact that now we are testing a lot of sounds, so the PCA space becomes very crowded and it's difficult for the distractors to be away from the target. We also tried with all the combinations, so if you have a longer training sequence, which is slide 24, and we observed that there is not a big change. Then we tried other experiments. Slide 25, experiment 2, we alternate with single sounds. So now we don't only have mixtures, but we also have single sounds alternated with the mixtures in different protocols. I'm going to speed up a bit because I don't want to take too much time. And then experiment 3 instead has some temporal delay. So we have the same number of mixtures, but we increase the number of single sounds among mixtures. So you see, for example, in case 0, there are only mixtures, but in case 2, we have 2 sounds every other mixtures. And if we go now to slide 26, you see our results compared with the human performance. So for experiment 2, case A and C are both above chance level, similarly to the human performance, while the biggest problem of our model is that case B, it's above chance for our model, while it's not for the human performance. And this can be due to the fact that when you are presenting sounds isolated, our model is picking up features that may be in the mixture sounds as well. And those they can distinguish it, while in reality human listeners cannot. Instead, for experiment 3, we have quite a nice reproduction. So as the delay between mixture presentation increases, also the performance degrades between our model and the human performance. So this concludes our reproduction of the experiments done in the original paper, but we also extended this pipeline to other input sources, starting with sound sources in slide 28. So you may remember that I mentioned that in the human experiment, they didn't use natural sound because it was a trivial task for people. But our model doesn't have any familiarity with naturalistic sounds because it's just randomly initialized, basically. So we can extend the experiment and see if we can perform the same task with sounds that are more complex. The only issue is that we cannot have a rigorous pipeline to build a distractor, because if you remember, we were using a Gaussian distribution to build a distractor, and here we have harmonic sounds. So we're just going to see a qualitative evaluation. In slide 29, we see an example of how the spectrogram of some naturalistic sound look like, and how, so we have the two single sounds on the left, and then we sum them, and you see a summed signal on the right. This is the mixture that we are presenting in training. And if you go on slide 30, you see a very similar plot to what we've seen before. So if we only have one mixture, then the network response is not specific for the different sounds, and the two targets and the incorrect and the distractor, which is just another naturalistic sound in this case, they all fall within the same cluster. But if we look on slide 31 and perform the same experiment with 3 mixtures, now there is quite a nice distinction of the cluster. You can see that with the naturalistic sounds, the clusters are more compact and they're also far away from each other, more than the synthetic sounds. So this means that the task is easier for the network. And then on the F panel, you see that the distractor in orange falls very far from all the other clusters. So this means that the performance of the network is basically perfect. And we did a similar experiment extending to visual stimuli. So on slide 33, you can see a brief introduction. The idea was not, let's say, risky, because also when we are performing experiments with sounds, we are treating them as images, so we can use exactly the same network. However, well, we have to tune a bit the parameters, but it's the same pipeline. However, the result is non-trivial because the distribution of the intensity within the image is different from spectrograms to naturalistic images. Fortunately, this worked. So on slide 34, there's an example of mixing two images, such in the same way as we do with the spectrogram. And then on slide 35, we see the example using these 4 images on the right side of the slide. And then you see also here, circled with the black circle, that different neurons respond differently to different sounds. So we have quite a nice distinction also among the presented images and the distractor, which is in purple. And slide 36 shows that you can obtain the exact same results also by changing images, just an artistic choice here. And to summarize, so I'm now on last slide, 37, what we've done is to exploit a learning rule for dendritic models to build a very, very simple computational model of the auditory system, which we applied to see if this model was able to reproduce results of blind source separation through repetition. So beside the experiment that I've pointed out, most of the protocols that we tested gave quite a good reproduction, maybe not in absolute value, but in chance level versus non-chance level. And also using a computational model allowed us to extend some result, make maybe some prediction compared to the human experiment, because we just have to run a code and we don't need to take a lot of people in the lab and do experiments. However, there are definitely some drawbacks of our model. First of all, if you remember, we are flattening the spectrogram. This means that we lose information about the temporal structure, and we present a sort of static stimulus for 400 time step. So this is something we should work on and take into account in a following project. Also, we have a spiking core, but we have a lot of processing and post-processing that are not spiking. So it would be also good to have a different way of performing, for example, PCA, which is spiking based. And talking about PCA, having this two-dimensional representation when the task becomes more complex leads to performance degradation. So another, we could exploit other ways of doing dimensionality reduction that don't have these side effects. But just to summarize everything, we have a self-organizing network because of how the learning rule works. So it would be really great to have some collaboration with hardware groups that are looking for models to do audio processing tasks in neuromorphic hardware, because this would alleviate some of the problems associated, for example, with doing error-related backpropagation. So with this, I'll wrap up my presentation. Thank you very much for the questions you had during the presentation. If you have any other comment, suggestion, or question, I'm really happy to discuss.
Thank you so much for your great presentation. This was such an interesting work, yeah, you did a really amazing job explaining it. Frank, please go ahead. I'm sorry. Go ahead.
No worries. I have kind of like 3 different questions. And first of all, I'm a social scientist and an audio engineer, but I'm not as close to this neuroscientific setup like Wissam is. So I still have a little bit trouble how these neuronal activities have been observed. You said experiment a lot of times, and I didn't get what is the empirical data in here. It put into what type of model? That would be my first question. I have two others.
Okay, sure. So the empirical data that we want to reproduce are data done on human experiments. There is no recording involved. So if you go to slide 6, you see a very simplified version of the protocol. You present the listener, the subject, with some mixture, either 1 or 2 or 3, or depending on the protocol. And then you present a single sound, which is either one sound that the person has listened to in the mixture, or a similar sound, but that was not in the mixture. And then the person has to say, this sound was in the mixture or not, and has to reply, sure, yes, yes, no, or sure, no. And then they do this with a number of patients, I don't remember how many. And then based on that, they build this ROC curve that we also use for our pipeline. So there is no neural recording in the human experiment. Instead, in our pipeline, we have to find a way for going from the network activity that we have run to the yes, no, sure, yes, sure, no. So if you go in slide 19, our empirical data now is the leftmost plot, the one that you have, in which you see the neural activity per neuron as a response to different sounds. And then all the pipeline, so let's say the novelty of the work, it's a way for going from that response to a yes, no, sure yes, or sure no response, which we've done. So basically, the neural recording are just simulation in our experiment. Does this answer your question?
Yeah, got it. Thanks. So the next question would be, can you, with this work, kind of differentiate between how an animal model would work and how a human model would work? So let's say the difference between the reactions of mice or something like that and humans.
I see. That's definitely an interesting point. But I think, so for how our model works, it is so simple that it's very, very difficult to say this is a model for a human auditory system. It is like a very, very basic way of encoding sound and then decoding into a classification spike, a classification output, sorry. To be honest, I don't know much about the animal auditory system. I think
You cut out there, Georgia.
Are you with us? Georgia?
Seems like you got in a weird matrix. We don't hear you anymore.
I think our speaker might have cut out for a second. She said that this would maybe happen if there was a power cut or something. Katerina, you can't hear anything, right?
No,
Hey, Jamie,
I
can you
can.
guys hear me?
Yeah,
Yep.
we can hear you.
Yeah, I think
George,
Clubhouse is acting up again. It dropped me for like 5 minutes
oh,
a
it
couple
did?
minutes ago.
Oh.
Oh, right.
I'm sorry, I've
Georgia,
lost
if
you
you
for.
can hear. Oh,
Oh,
now we can
she's
hear
back.
you.
Oh, she's back. Hello
Yeah,
again.
Okay,
Yep,
okay,
hi.
sorry. Can you still hear me?
yeah, yeah.
Okay,
Thank you.
Yep,
perfect.
you're back.
Sorry, did this answer the question? I stopped hearing at some point.
We didn't hear your answer. We just
Oh,
heard the question.
okay, sorry about that.
I'm sorry.
I'll say it again. So I was saying that I am sure that the animal system has some way of performing blind source decomposition. Because, for example, If you think of an animal in danger, it needs to be able to distinguish like the predator that's approaching from the background of the river that is flowing, for example. But I am not aware if there are similar experiments done with mice. For sure, it's more difficult to get a verbal response. I mean, it's impossible to get a verbal response. Maybe you can decode some behavior from it. But another point that I was mentioning is that our model is very general. So it's not, we use it to reproduce experiments done on humans, but it is not a trustworthy reproduction of the human auditory system. It's just a, let's say, a very simple way of going from an acoustic input to a classification output.
Yes. Okay. So thank you. That kind of all of these answers lead into my last question, and it's interesting because I'm doing in my free time kind of an quite similar experiment, not to predict something on a neuronal level, but towards recognition and focus of meaning in multi-speaker settings. So this kind of cafeteria situation where there are multiple parties speaking, I do wonder what to what extent these just similarly to what you are doing here, to what extent not only the recognition of different voices could happen, but also how much of the differing factors could be recognized. So I would be curious what do you think on the recognition of not just tones, but actually the words. What would you expect with what you've learned so far from your experiment? How much could any meaning be recognized, and to what extent do the factors volume and pitch, notes, spatial position and distance from the speakers play into the possibility of recognizing a content of words, if you could speak to that?
Mm-hmm. So that's a super interesting point. Thanks for bringing it up. So in terms of features of the sounds within this task, if you look, for example, at slide 29, um, if I'm not wrong and I remember correctly, the top left figure is speech. So you see that the frequencies that are high intensity are mostly those towards the low range of the scale. But this works as well when you have like chimes, which would be the bottom left figure, in which you have quite a different structure. So the model is general enough to work both on these highly structured sounds and those synthetic sounds that we've seen at the very beginning. So this is not an issue. But in terms of speech recognition, for example, we would need to implement a different pipeline. So we would still be able to use the network in the way it is, so using the input neurons and then the output neurons, but then we would have a different decoding structure. So for example, if you go on slide, if you go on slide 19 again, you would have the first step, but then, for example, instead of doing PCA to perform classification, you may need to use a different way to do readout from the neuron responses to relate the neuron responses to words, for example, which I have not done, but it could be an interesting extension of the network.
All right, yeah, thank you so much.
Thanks for the question.
I wanted to just make a quick comment and then a follow-up question.
Sure.
On the question about the animal, like doing experiments for this in animals, This would actually be pretty interesting to do. I thought about playing around with this, but I haven't. Um, like you said, you could do a behavioral response. Train the animal to demonstrate that it can pick out maybe a sound in a mixture and do the traditional psychometric function. But you could also apply the method you did to your network to the neural data. In other words, you could make a classifier over the neural responses in the same way.
Yeah, that's a great idea,
So,
actually.
yeah, so this is pretty popular these days, neurometric versus psychometric functions. And it would lay over perfectly with what you're simulating. I was wondering though about you did the classification using the mixture model in 2D, so using the first two principal components. I'm curious how much variance was explained with the two dimensions and whether you wouldn't do better at classifying in higher dimensions.
Mm-hmm. Yeah, so I have actually the number on the paper. Let me look for it because I've measured the variance. Can you still hear me?
Yeah, we can hear you.
Yeah,
Okay,
you're fine.
perfect. So the variance that we explain, it was pretty high, but just give me a second. I don't remember the. So not too high, but it's about 40% of the entire annular response. So a consistent percentage, but we are losing a lot. So definitely this is one of the drawbacks of the model, which wouldn't be too hard to fix,
Yeah,
actually.
yeah. To me, that says if you added just one more dimension, you might be able to separate these clusters pretty easily.
Yeah, no, you're totally right. It's something that the reviewers suggested as well, and then we decided not to change too much the pipeline. But it's an excellent point. Thank you for commenting with it.
Um, yeah, and thank you so much for this amazing work. I wanted to ask you if you are collaborating with. We had here Dr. Guat from IBM too. I don't know if you know him.
No,
And
actually
then,
not. What's his name? Sorry.
let me check again how to spell it because I maybe said it wrong. But anyways, the question was if you are looking into next to implement something like synaptic plasticity modeling to basically have some sort of excitability changes to keep the memory basically of different sounds, or is that something that is really out of your scope to do that?
That's actually something interesting. But no, right now I'm not working on this project anymore. So we've wrapped it up with the paper, and I'm not looking into particular extensions right now. But if ideas come up, why not?
Oh, I looked him up. It's Agazi Sawat. I don't know if you
Yes,
know.
I think is in my office. Is at IBM Zurich, right?
Yes. Yeah. He was here like a couple of months ago.
Interesting. Yeah.
Because he looks at in-memory technologies for deep learning acceleration.
Yeah, that's him. They have a very interesting hardware, but I would say that it still needs a bit of working to get to these tasks that are a bit more complex than the benchmark ones that they're testing now.
Yeah, great. And so you mentioned that you can basically also use a visual input and have similar, like you can use this design to also process visual inputs, correct?
Yes, that's correct.
Are you thinking of someday applying this maybe to a sense of smell? I know it's really hard to process sense of smell information, but I read there's some, there's a lot of effort to having accurate sensors. Do you think, you could also process that type of data just as well?
I think it wouldn't be too difficult if the encoding is done in a similar manner. So I'm not too familiar with olfactory sources processing, but I would say that like when you encode them, each, let's say while with the sound, we have different frequencies, and then you have one pixel and then one neuron per frequency and time frame. I guess with olfactory processing, you would have molecules instead of frequencies. And then if a molecule is present, you have a spike. If it's not present, you don't have a spike or something like that. I'm not 100% sure that this is how it is done. But if this is the way, then in principle, I wouldn't see a reason why the pipeline wouldn't work.
That's exciting. So in theory, you could process any sensory information and distinguish better between different inputs. Also, let's say tactile processing for, let's say, artificial skin. Maybe people that lost their sense, in ALS, and so that lost the ability to move, and paralyzed people. So that would be quite amazing if your approach would be better at processing artificial sensory inputs, basically for people that have different, lost different senses.
It would be really cool,
I
yes.
think that would be,
actually, we didn't think about it, but now that you mention it, like obvious, you can do all the senses.
yeah, that would be amazing. I wish, I hope you'll do that, because, it would help people so much. And if we could translate it into, an input for people in another spot. I'm thinking about Miguel Nicolili's work, where he created an exoskeleton, and that had basically sensors where the feet usually would be, and created basically a tactile input on a region on the skin where people still had sensation, like on the arms, for example. If they were just paralyzed from the legs down. So you could basically, if people like in ALS, and so gradually lose their kind of senses and their controls of movement, you could have type of an exoskeleton that feeds that in, let's say, into the neck, or wherever that would be, or maybe even directly into the brain with some chips. That would be amazing to have the input, because we had a room here where researcher where we had different rooms where researchers were good at reading out basically what is happening in the brain for speech, like to fasten up speech, and locked people that were locked in, or imagery. But the other way around, I think, is still lacking, and that's where you come in. So that would be really cool.
It would be super interesting. Yeah, I mean, with sounds and images, it's easy because these are data sets that you can either build very quickly by, I don't know, looking up images on Google, or looking at sound recordings that you have on your phone on their public database. I'm not sure. I think olfactory and tactile are lagging a bit behind. Maybe now there are some common benchmarks, but I guess when you have something that's so easy to try your model on, then you go with the easiest way. But for sure, it would be super interesting to see if it also works for definitely for olfactory, because you can have superposition of different smells, different odors. I'm not sure tactile how you could combine two tactile perceptions. Maybe like you can combine, I don't know, temperature with texture, something like that. But yeah, something to write down and keep in mind.
Yeah, maybe just from two hands, you know, from both sides, like the same object, but from the opposite side. So I don't know if that would be enough. Yeah, to distinguish, probably not, but yeah, maybe two type of textures. That would be really interesting. Yeah. So yeah,
Just
I don't
wanted to
know, maybe. Yeah, go ahead.
I just wanted to, I have to run off to a meeting, unfortunately, but I wanted to thank you for the talk and presenting your work. There's a massive overlap in our interests. Clearly, I study biological dendrites and build models of dendrites, single cell, and networks. And I came into neuroscience from the auditory field and spent a lot of time thinking about this blind source separation problem. So super interesting for me. I just wanted to comment on the reason I was so interested in this cocktail party problem, as they call it, is it sits inside of this larger blind source separation problem. And general solutions to this problem would affect every field. Any field that has a dimensionality problem at a sensor, which is every field, would benefit from a general solution to this. So really, really cool stuff. I'm in the Boston area, and I know some of. I heard you say something about working with Gabriel, but would be curious to see what you're up to these days. But gonna have to take off right now, unfortunately.
Yeah, let's catch up. I'll be in Boston in a month, more or less. So I don't know if
Okay.
you can see my email somewhere or give me your name through message. I'm not really familiar with the app, but it would be really interesting also to know what you're working on.
Sounds good. All right. Thank you for your presentation.
Thank you for your
I
questions.
can always connect. Yeah, I can always connect you too. Oh, you're going to Gabriel's lab to work?
Yeah, so I was
I don't
working
know.
with Gabriel. I mean, I'm still working, but now I'm working from Switzerland,
Okay.
and I'll be visiting again
Yeah,
this summer.
great. That's amazing. Yeah, I can always connect you through email, so please let me know. Frank, you unmiked. Frank, you can go ahead. I'm sorry. I saw you unmiked. Okay, maybe we lost. So yeah, I wanted to check in. Oh,
Can you hear
yeah,
me?
now I can hear
Can you
you.
hear me
Yeah.
now? Okay, so my app is so.
Now
I
you're
think you're
muted
unmuted.
again. I'm sorry. Yeah, the app is kind of glitchy. I'm sorry about that, Georgia.
No, it's fine.
Frank, we cannot hear you again. Maybe you wanna leave and come back in really quick. Sometimes that helps.
I'll just
Ah,
do that.
now it works. Now we hear you.
Oh my goodness.
We hear. Can somebody else hear Frank, or is it just me?
I cannot.
I hear him come in like for a split second and he's gone again like that.
So
But now he's
I
gone. Oh,
Yeah,
there he
yeah,
is.
yeah, yeah.
Back.
I resetted the app. This app is so super glitchy as of the late updates. I just wanted to point out that Marty, who's on stage, and Pierre, who is in the followed by speaker sections, they both wrote questions into the chat. So Marty, if you want to ask your question, you should just unmic and go ahead. And maybe Pierre also wants to come up. I just want to point that out because it's in the chat, and I thought it was on the topic.
Thanks for mentioning. I didn't check the chat. I was too focused on the audio.
Yeah, no, it was my job. Thank you, Frank, for mentioning it. So, Madi, do you want to ask first, and then we'll ask Pierre's
Hi,
question? Thank you.
thank you very much. I have a question that is there any implementation of your model on GitHub or other repository?
Yes. So if you open the paper and just do Control F
I'm
GitHub,
reading the paper
it comes up.
actually.
So I mean, I don't remember which page is it. Let me just do a quick search. But yeah, there is to reproduce one of the experiments, but then the code is the same. So it's page 17 in the data availability. There is the code.
I see. Yeah. Another question that can I use this actually DAN or ADN for implementing generative models such as GAN? My means is, can I make a generative model such as GAN models, but instead of using usual neural nets such as perceptron, I use ADN neurons. Is it possible?
So I guess they are a bit too simple for GANs. And definitely you would have a different way of training them because you need a generator and a discriminator. So definitely with this specific model, no. But I'm not sure if there are some other models that are spiking and that there are generative. I mean, spiking is a bit behind the progress of just general ANNs. So I wouldn't be surprised if they're not there yet, because it's much more complex to train this kind of network. So as you can see, we only have one layer, while GANs are much more complex and deeper. So I'm sorry, I don't think it's the field is there yet. But please let me know if you find some other dendritic neuron models that can do that.
So it's not proper for applications such as TTS, I mean, this is text to speech, or generating speech from other features.
So there are some spiking models, but that are non-dendritic, so just one compartmental. There is a postdoc in my lab that's working on that, and the type of model is called SNU, which stands for spiking neural unit. But that is trained with backpropagation, so with supervised signal, and can perform speech processing. But I'm not aware of models that work with dendritic neurons to do that.
If we use Fourier transfer of the wave instead of using the signal directly, you think how will be our result? I mean, this FFT is good pre-process for this model or not?
That's a question that unfortunately I'm not sure I can answer. I mean, I don't know what the answer would be. Yeah, no, I'll be honest. I don't know.
Okay, thank you very much. I'm done
Sorry.
my
Thanks for your
speaking.
questions.
Just to highlight it, the model is in that linked paper on page 17, and it's on GitHub Georgia D and then / dendritic neuron BSS. So that was on that model that is in this paper.
Yeah, thanks for pointing it out.
So, Katarina, I don't know. I have a hard time to access the chat without without having to wait a minute to access it. So maybe you could read out the question of
Yeah.
Pierre.
Yeah, thank you, Frank. So Pierre is asking if you could use this for emotion detection. For instance, can you imagine that the loss of temporal info is actually a plus in that case because you could overlap various levels of emotions in the same spectrogram and make it easier for the model to find clusters? Can't jump on stage, but thank you very much. And
That's a very creative question. So we've reviewed all the different sensory senses. So emotions, it's very interesting, actually, a bit unexpected. So I've not thought about it. I'm a bit pragmatic. So when you want to use this kind of model, it's good that you have some signal. I am not sure if you're talking about emotions in general or like decoding emotions from, for example, behavioral data. I don't know, you have a collections of data that are sweating, smiling, loud voice, or other factors that can relate to emotions. If you have something like that then you can encode into some numeric form, which then can lead us to have spike trains, then I guess you could do that. I'm really not familiar with the field, so I don't know, maybe there are some computational works that analyze emotion in a quantitative manner, which if there are, I would be really interested in knowing more. So again, just short answer to the question, if you can put emotion into some matrix or vector, I guess that would be doable, yes. But my question for you is, do you know if there are some ways of encoding emotions into numbers?
Hi, Georgia. May I ask, I was reading your paper, the part here, now I wrote down the question because it's a long one. When non-negative matrix factorization is useful in extracting spatiotemporal patterns within audio, I was trying to understand that part a bit better, but with regards to that, does that mean it could separate all the separate notes, say like played on a guitar if someone strummed it, or if there was even an orchestra all playing things together? Would it be able to separate all of these things and hear where everything's from, what pitch it is, what note and everything?
I see. So non-negative matrix factorization is another way to solve the cocktail party problem, which has been observed to be very well performing lately, because before they used this independent component analysis that had the drawback that you had to have as many sound recordings, as many sources, and with non-negative matrix factorization you don't have this constraint. So I'm not an expert in here. I guess that you can apply it into general context. So what you were mentioning, for example, in the orchestra source separation. So this is, let's say, a parallel algorithm to what we are implementing. We are doing like an alternative solution. Is this maybe I missed a point in your question, or these answers?
No, okay. What I was just wondering is if it was when you were describing and this, it said it was quite effective in music, and I was wondering it could have been, but I was misunderstanding the part because it had my mind thinking towards if it could separate sounds like that, if a model could be made so that computers, machine learning could actually learn to play music using this model, understanding the balances between sounds and how they're all relating to each other. If a machine learning thing could, you know, adjust things and understand how the balance would go. That was just my simple musings, but sorry if I was missing the point.
No, it's interesting. I mean, just a more general comment on my side. These kind of models and algorithms are very task specific. So they are basically, they contain a set of parameters, these weights that you train on a specific task to do a specific job based on that data set. And it's very challenging to make them generalize within the same task to a data set that is only slightly different. So using an algorithm, exactly the same model that you use for blind source separation, I would say that maybe you exploit similar mechanism, but you cannot apply the exact same network to perform something else, such as learning music. There are, of course, points in common, but you need every time to retrain something on the specific task and dataset you want to work on. So again, maybe I missed your point again.
Thank you. No, look, this is my error in because what you're talking about is absolutely fascinating and quite complex, and I'm very, very impressed at what you're working on. So thank you very much for indulging my thoughts on my questions. Amazing work.
No, of course, it's interesting to see all these points of view. And thanks again for the questions.
Yeah, thank you so much. Are there any last questions or comments? If not, the idea has been going on for over an hour and a half. Dean, do you have a question? Go ahead.
Yeah, I guess I do. You say that I realize that this is a simple system, but yet it is flexible enough so that it can be used on visual data, even though it was created to process audio data. So would it be possible to modify it such that it processes spoken language in order to determine which language it is that is being spoken? I don't talk about translational interpretation, just determining which language it is.
That's actually a very cool application that one could think of. I haven't tried it. What I could say is that for sure you can extract different features from different languages in terms of the spectrogram. So for example, in terms of frequency, maybe pace at which the different syllables are coming one after the other. So there are some temporal features that the model could detect. I am not 100% optimistic that then this would work very well, in the sense that you would need a very large training set. And the model right now, as you maybe you remember from some of the figures, when you have a large number of sounds or a large number of mixtures, then the performance of the model was decreasing with the complexity of the tasks, which would be a bit incompatible with having large training sets that you need, for example, for language detection. But with all the modifications that we were thinking about, for example, not using the PCA, but using multiple dimensions, or using different ways of decoding the signal, I mean, I'm not saying it's impossible. I think definitely it requires a modification, but the core, in principle, detects temporal features that are that could be related to language features. So after this discussion, now I would need like 2 months to apply all the suggestions that you gave me, which are super interesting.
Yeah, thank you. Yeah, I hope we didn't.
Thank you.
We didn't overwhelm. Obviously, it's really easy to suggest things, right? It's hard to do the actual work. That's the easy part.
Maybe not hard, but definitely time consuming.
Yes, exactly. So, yeah, whoever made suggestions, me included, please offer some help
That would be
so we
great.
can get those done. Exactly. So, yeah, let us know, reach out to us, and then maybe we can all help and publish a paper together. I don't know.
That would be nice.
Exactly. So, anyone that wants to reach out and help with implementing those ideas, let us know. Gilbert, you on mic? Okay, yeah. So thank you so much for offering so much of your time and to share this really interesting research. You can see that it sparked a lot of ideas in us. So we are really glad that you came to, to answer all the questions and to listen to our ideas. So yeah, thank you everyone for coming and for asking questions, for interacting. And Georgia, as I said, reach out, tell people what to do. And I hope you'll come back one day and give us updates what you do on your next, chapter of your life. And congratulations, and we wish you all the best for your future.
Of course, first of all, thanks again for inviting. I really appreciate it. It was such a nice chat with a lot of ideas. I'm really grateful for the opportunity. Thanks again. And then I mentioned it already when we met last time. I'd really love to join as an audience. One of the next rooms. I've seen there are some super interesting topics. So yeah.
We would,
Oh yeah, feel always welcome to come and to speak. So if I see you, I'll invite you to come up to the stage and participate in the discussion. That's wonderful to hear. We are always happy to hear if you find this interesting. So thank you.
With pleasure.
yeah, we'll definitely welcome you for sure. And normally we would, I would wish you good luck, but this is such remarkable work here that you don't need anyone luck in getting your PhD and coming to us again as a doctor. And thank you so much for your time and your incredible work.
Thank
Thank
you
you.
so much for your curiosity. Very appreciated.
Wonderful. Okay, we are looking forward to seeing you, hearing you again.
Yes, yes, yes.
Maybe you can use Clubhouse one day for continuing. But yeah, and thanks everyone. If you like discussions like these, follow the club, become a member of the club. And our next room is tomorrow. Dr. Kihi, who will talk about self-assembled logic circuits based on proteins. So that will be really interesting. And then on Thursday we will have Dr. Q, a more climate room, talking about the expansion of the desert climate in Central Asia. And on Friday we'll have Dr. Cagan talking about gene mutations across species that shed light on aging, solving aging maybe one day. So yeah, thank you again, Deirdre. It was such an honor having you here. And yeah, we'll hear you all back soon. Thank you, everyone.
Thank you. Bye.
Bye.
Thank you.
Thank you, everybody. Bye bye.
Thank you so much. Goodbye.
3, 2, 1. Bye, everyone.